Further, we will study about Volvox, the new genus, belongs to class Chlorophyceae, order Volvocales, family Volvocaceae. Volvox is the most highly evolved synovial algae, both in its morphology and reproduction. It has about 20 species which occur in fresh water of temporary or permanent pools as a miniature spherical or ovoid ball of pinhead size. The colony multiplied so rapidly during rainy season that the entire pond is covered by them and look greenish. The genus represented in India are the Volvox globator, Volvox aureus, Volvox africans. They are the fairly common found in the ponds. Thallus structure. Volvox is a colonial algae, and colonies are oval to spherical in shape, each with a 500 to 60,000 cells. The cells are biflagellate and are arranged in a single layer within the periphery of gelatinous colonial envelope. Each cell of the colony has its own gelatinous sheath, and the sheath of the individual cell may be distinct or confluent. In most of the species, adjacent cells are connected by plasmodesmata strand, which are formed during cell division. In some species, in Volvox monani and Volvox tertius, cytoplasmic strands are absent. The central hollow cavity of the colony is filled with water or gelatinous in case of Volvox aureus substance. The cell in a colony are usually pyriformed in shape with a broad posterior and narrow anterior end. They are eukaryotic and mostly shows a typical chlamydomonas structure, but in Volvox glabrata and Volvox rosesti, it is a spherula type. The protoplast is enclosed within a semi-permeable plasma membrane which form the outer boundary of the cytoplasm. There is a single nucleus, a cup-shaped chloroplast, and two contractile vacuoles, two flagella, and an eye spot in each cell. The nucleus is connected with the flagella through the neuromotor apparatus of the cell. The eye spot is toward the external face of the cell. It has a colorless biconvex photosynthetic lens and a curved pigmented plate. The massive cup-shaped parietal chloroplast occupy the posterior border part of the cell. It has a single pyrenoid made up of a central protein core and a peripheral starch plate. Most of the colonies are vegetative and the only few are reproductive. The reproductive cells do not have flagella and they are relatively larger than their vegetative cell. There are only a few or so reproductive cells at the anterior end of the colony, whereas the posterior end has many reproductive cells. The cell at the anterior end of the colony have a relatively large eye spot than those at the posterior end. Thus, the colony of Volvox shows polarity. Reproduction. Volvox reproduce by asexual and sexual method. Asexual reproduction usually takes place during spring and summer month, whereas sexual reproduction starts toward the end of the summer season. Asexual reproduction. Asexual reproduction takes place by a special type of cells situated at the posterior end of the colony. These cells are called gonidia or parthenogonia, are about 10 times larger than that of the vegetative cell. They are spherical and highly vacuolated. They develop numerous pyrenoids within their chloroplast. The nucleus is suspended in the center of the cell. The gonidia do not have flagella and eye spot. Usually two gonidia are separated by several vegetative cells. A gonidium is embedded in a hollow gelatinous cavity of the colony and along with its globular gelatinous sac. The protoplasm of the gonidium divide and form daughter colony. Development of daughter colony. The daughter colony developed from the gonidium. The enlarged and the spherical gonidium undergoes their three longitudinal division with respect to the colony, forming 8 cells. These 8 cells arrange themselves in a curved plate, and this stage is called the Planck stage. Each of these 8 cells again divide longitudinally, forming 16 cells. At this stage, the cell formed an almost complete sphere except for a small opening, the phyllophore, directed toward the periphery of the colony. The phyllophore eventually become the posterior pole of the daughter colony. These 16 cells divide repeatedly and form as many as 60,000 cells. Large number of divisions usually do not occur, and divisions stop after a fixed number of cells have been formed. At this stage, cells are naked and are in a close contact with each other. The anterior papillae part of each cell is directed toward the center of the hollow. The normal orientation of the cell of a colony is achieved by the inversion of the cell of the sphere. The inversion starts with the formation of a constriction at the posterior end of the sphere, and that is opposite the phyllophore, which pushes the cell at the posterior end toward the phyllophore. In the meantime, the phyllophore becomes broader and the cell at the posterior part are pushed toward it. This process is known as inversion. At the end of inversion, the anterior papillae ends of the cell faces toward the periphery of the colony. After inversion of the colony is completed, the cells secrete a gelatinous cell wall and develop flagella. In this way, a daughter colony is formed inside each gonidium. Many daughter colonies may develop within a parent colony at the same time, and they swim freely in the gelatinous matrix of the parent colony. The daughter colony are released after the rupture or disintegration of the parent colony. The parent colony usually degenerate after the release of the daughter colony, but in Volvox africans and Volvox carti, 2 to 4 generation of daughter colonies can be found within the parent colony. Thank you.